Everyone, welcome.
And set and drove a lot of the new data that I'll show you today. We'll give you a big update on that. Lots of new data there in terms of panel design, high parameter. And so we'll talk about some of the foundational technology first and then jump right into that. And so please make it live and fire those questions away, because that's just a lot more fun. So I figured this is really what we've really been spending the past, at least quarter doing the past 3 months. We've been spending an exponential time looking at exponential graphs. And so I thought today should really be no different. But we're going to take a more comfortable exponential graph. This one, we're looking at colors, the number of colors we have in flow cytometry. On the y-axis, a plot many of you will be familiar with. And on the x-axis, we have time. And there's some interesting observations I want to pull apart from this relative to where we are right now in flow cytometry. And then Mario, as is his wont, named this Roeder's law for flow cytometry, where the number of colors doubles every 10 years, right? And for those of you, I can see there's actually folks on here who are even outside of flow. For those of you who are dialing in, this is essentially our version of Moore's law, and it's quite interesting. And so, these are projected for dyes currently in development. Although I think we've seen that some of the brilliant blues are a little bit slower than what was, I guess, anticipated versus the fact that we already are in 2020. But I want to look at this graph a bit of a different way, right? Which is, and you see these conjugates in here, is what has it done for hardware development? What's been this relationship in between dyes and hardware? And so if we go back here, you can see when you've got the development of some of these tandem dyes, it actually drove and pushed the field to create, for instance, the first 12 color flow cytometer with the 12 color Fax Vantage, right? And then when we got the quantum dots, it again pushed the field to then jump again and develop the 18 color LSR2. And in fact, dyes were really driving hardware development. They would make a leap. And we see this actually in the past several years, where you get the next leap really from the brilliant violet dyes, and you get the Fax Symphony, if you will, in response to that new capability, right? And so dyes have been really driving hardware development, and that's really been the universe that we've lived in. I would also add another point here, which is that they've driven democratization, because as you get these increases in the number of questions we can answer, you get democratization of the instrumentation down at the low and then the mid end, right? So if you look at something like the way that you can get an Attune or a Cytoflex now that can drive these number of color experiments, you get that democratization. You get a lot of different sort of benchtop instruments, et cetera. But where are we now? The answer is now we're in what I'm calling an inversion, where you've got instruments that are well over detectored, if you will, versus what we can do in terms of dyes. And a lot of that's driven through spectral cytometry, right? As you'll expect, right? You've got a 5 laser Aurora that has 64 detectors. The max colors that's been shown on that, at least on their website, is 40 colors. Something that we'll talk quite a bit about today. And then you have also the 7 laser ID 7000, which has 188 detectors. There's at least a press release out that says that they can do 44 colors. I haven't seen any data on that, so I guess we'll just wait on that. But we'll actually be showing some data again today to push that envelope. But I think the point is that we have so much latent capability across this entire spectrum, right? Not just the 64 detector instruments or the 188 detector instruments, but even if we take it all the way back down to, for instance, like an Attune or an LSR4 Tessa, in the way that we're using the current, for instance, BYG space, something that we'll spend a little bit of time on today, we're still underutilizing the nascent capabilities of our instruments. And so again, we'll spend some time talking around that today. So this is presumably called an introduction to NovaFluors, although, as I'm sure you'll appreciate, and it's probably the reason, at least half of the reason you showed up, is we're going to talk about quite a bit more, right? And actually quite a bit more in terms of actually how we can plug and play our labels into a backbone panel to achieve 40 colors, and then certainly our, ambition and drive and goal to go well beyond that, right? And so the above and beyond 40 is what are we doing to really share that, discuss how we did that in terms of panel design, in terms of methodology, in terms of what are the insights that we're pulling apart on that, and something that we'll spend a good amount of today on. The first thing is I always like taking a step back in terms of panel design and where we are, right? And so, we're thinking around panel design, we really have to consider 4 different things, right? Which is spectral overlap, right? As I've always said, some of these labels lead a decidedly more colorful life, right? We've got antigen density, right? Where, even when we think about antigen density and mapping antigen density, low antigen density to high brightness, we have fixed brightness, fluorophores, and so we're really sort of stuck there, something that we'll spend a good amount of time talking about, particularly relative to PE and some of these PE conjugates. We also have spillover spreading, which I think is really a pernicious issue in the sense that it has a huge impact on data analysis. And it's not that spectral cytometry makes this any better. There's no magic in that. When you have off-channel fluorescence, you still have contribution of spread, and you still have a difficulty of data analysis. Again, something we'll touch on, and we'll actually use that as a metric for looking at what's the performance of an ideal fluorophore. And then also staining index, right? So actually, can you separate biological populations? We've had a lot of conversations around brightness over these past really 9 months. And for us, what brightness means is the ability to resolve a biological population. And so we think a lot around stain index. We also think a lot about separation index, which takes into account actually the 84th percentile of this negative population. Again, something that we'll touch on, because really what you really want is not just the brightest thing in the universe that also has high background, but something that has low background and has really good separation, right? Because that's really where we want to drive towards. This is something that we'll discuss, but it's been something that we've been rather obsessed with, is if you have clean fluorophores, you can limit spillover spreading, you limit staining index. Could you drive to the point where you actually have high resolution biology? And this has been something that, again, we'll show you some data on, and we'll discuss that. But I think one of the other thing is, if you think about all of this, really we're dealing with fluorophores and really mapping them into our biological problem, not using fluorophores that are designed for biology. And dye performance and our compromises in dealing with dye performance informs, certainly it is the reason that we have 3 or 4 of these without antigenets. Antigenets is a function of the underlying biology. But 3 of these are dealing with the compromises of current dye technology. So we're going to talk around dye performance today. 7 lasers obviously out there in the universe, so to speak. No dyes really off of the deep UV. There are very few lasers out there, very few instruments out there that have a deep UV. The IR, again, similar story there. Very few instruments that have an IR. Very few number of labels out there as well. Really, we're going to focus our attention on these 5 lasers today, and in particular, focus in on the blue and the yellow or green lasers, an area that really has seen almost no innovation, right, in terms of fluorescence technology, for about 15 years. And so, why did we first start thinking around blue and yellow green, right? As I'll show you, our platform technology actually enables us to go after laser lines, if you will, at will. And one of the reasons was looking at these foundational dyes in our field, like PerCP, right, which is excited off of the blue laser, but then has cross-excited off of both the violet and the yellow-green, or something like PE, which has a primary yellow-green laser, but is also cross-excited by the blue. And thinking about these foundational dyes, if you will, as a highway, and thinking, man, we are driving brand new cars on really old highways, particularly with respect to the BYG dyes. And to characterize that a little bit better, and we'll be showing some of these ribbon plots today, and we've put out a white paper that has these ribbon plots for our labels as well, is introducing this, this concept is, here we've got these 5 laser lines with their different detectors, and we've got intensity here on the y-axis. There's a primary detector here. That's just noted with this bar shape. And then everywhere else that we have fluorescence, there's actually this introduction of spread, right? And so there is no magic around that. You will be introducing spread into your analysis. And so to look at this and look at these foundational dyes, for instance, that are in the blue, right, we've got PerCP that certainly has a decent amount of primary channel fluorescence, and has this signature, but really a signature is a function of a badly performing fluorophore, right? A signature is because it emits or is cross-excited by other lasers, right? And we can see that there are more guilty parties in that. You've got the brilliant blue dye, which obviously, when you look at these plots, emits quite a bit into the violet, right, and into the UV. Again, another area where we focus to say, look, really, if you have a blue label, it really should be silent in the violet, right? We'll leave purple rain to Prince. And it's very similarly, brilliant blue 700, obviously quite a bright dye when, with respect to the blue, but a huge amount of emission in other channels. And so let's take a step back from all of that. What if you could start from scratch? It sounds like a crazy question, and I agree. That's what got me interested in this in the first place. But having come from a world where I was designing software, and then looking at, for instance, algorithms for many years, and looking at we would always say like bad data in, bad clustering or bad results out. But then you think, well, really, what's that all based on? And the answer is, well, it was really the fluorescence, right? And so given that, what would panel design be? And so the 3 requirements that we've captured And interestingly enough, really a lot of these were a result of a lot of different great customer conversations we had, particularly this middle one, consistent and stable. But the first one is one that we always pick out, bright and clean, obviously, right? But notably, also silent in the violet, right? Can we protect the violet space, right? If we're going to use multi-parameter, if we're going to really push multi-parameter, we really want to use all the capabilities we have in the violet. We certainly want to limit also laser cross excitation. What if we designed for that? The second one, as I mentioned, which was definitely an emergent one for us, which was this notion of how consistent, how stable can you make these labels, and how can you push that? And we'll talk quite a bit about that, and I'll show you some brand new data and how we're thinking about consistency. And the third one is a brand new capability, which is controllable brightness, where you can actually choose brightness based on an antigen density or panel. And again, I'll show you some brand new data on that, where we can actually tune the brightness stoichiometrically of our labels up and down. And so how do we do this, right? What is the platform that lay underneath all of this technology? And so what we do is we take single-stranded oligonucleotides, and we bind on small component organic dyes. And then what we can do is anneal them and construct this phyton structure, which is a cruciform. And what that enables us to do is engineer down to the sub-nanometer level the way that dyes can interact with one another. And then we can take that phyton structure and then put it onto an antibody, because we can take part of the arm of this phyton and extend off a single-stranded oligo. Had some really interesting email conversations, for what it's worth, some interesting questions about this over the past couple of days. And a lot of people have been asking about tandems. These are not tandem dyes, right? But they do rely on FRET. They do rely on fluorescence resonance energy transfer. And what we're able to do is engineer the distances of the way that these component dyes are interacting so that you have incredibly consistent and dialed in FRET. But they are not tandem dyes, right? Because I can put that on any antibody and the space is fixed, right? And they're sitting there on the DNA, both spatially, right? Because, and that's important because FRET actually scales with the square of distance. But also, if you think about the way that you're currently using your tandems, that is exactly the same FRET every single time, right? So you don't need to run single stain controls that are specifically that antibody, right? That rare, CDX marker that you then have to run on beads, because the FRET that we're using here is consistent every single time, right? So you could run CD4 as your comp control, whatever you want to do, right? And the platform enables us a couple of different other lifts that we'll talk about. The first one is we're relying on standard oligonucleotide manufacturing to make these labels, right? And so you'll see that leads to important pieces, important data points around consistency. The second thing is it gives us the capability to do tunable excitation and tunable emission because of what I've described with respect to these component dyes. And the other part of this is tunable brightness, again, something that I've already mentioned. We've also done some tunable signatures. I'm not going to be touching on that today. There's a lot of biology really that stands between us and the ability to leverage tunable signatures, but it's something that we have done. We actually have 4 different tunable signature dyes where we're able to create spectral signatures. Not an area we're going to focus on today, because we want to drive towards this high parameter biology. So let's focus on this first one for a second, which is this notion of bright and clean. What does that look like? So these are the first 6 fluorescent labels. So just a moment on the nomenclature of our labels. Blue is the laser line that they're excited off of, and the number is where their peak emission is. So again, honestly, just making a panel design piece of this easier from a nomenclature point of view, and then, as we'll discuss much later, much easier from a spread and from a performance point of view. But I think what you can see immediately is that we've separated the blue and the yellow-green excitation. So the way we've thought about this is actually stacking these blue and yellow-green laser lines, right? And again, we can do that off any instrument. It could be a Fortessa, it could be an Attune. We've done a lot of development on the Attune. And then also, as we'll show you, it can be on a 5-laser Aurora, right? So this maps into a number of different types of instruments. Again, what you can also see here is that we're getting very limited cross excitation as we talked about between these lines, and we're able to step the Stokes shift out. Again, that's for these first 6 labels. And these are the ones that will really carry through the rest of the talk and talk around panel design and some insights around how they interact with other labels that you might be using. We've taken these spectra too from, because from an experimental planning point of view, you want to see the spectra for sure. We've taken them and published them up on FPBase, which is this open source spectra viewer. Great resource there because you can look at it with in vivo dyes or other fluorescent labels. The folks at Cytec also were kind enough to take our labels and publish them up on the Cytec full spectrum viewer. So not only can you see the spectrum, but you can also look at similarity matrices with other labels. You can also look at complexity in terms of, okay, well, if I add these labels, what does the complexity look like? And again, we'll comment actually on both of those items in a moment. But I think if we look at this plot in the blue and yellow green, it gives you a sense of what we're up to, right? We're hitting these detectors very, very cleanly, and we're able to stack, right? And we obviously have some room to grow, as it were. And we're also very quiet in the violet, right? We're not adding spread to the violet and the UV, right? Or really even much spread into the red, as I'll show you. And again, we'll show you that actually with our high parameter panel development in terms of what is the impact of adding it at really at the very edge of what's possible, right? In terms of high parameter, and how much spread is this adding in a fully loaded panel. And I'll show you that actually iteratively later.
Okay, Mike, I'm going to just interrupt real quick. We've got two questions.
Cool.
One is, why would you want to do a 40 color panel? You know, I think to some extent that really goes to, you know, maybe a limited clinical sample, you know, in terms of commenting on that. And then second question is whether folks can conjugate these to their own in-house antibodies. And we do have a conjugation kit available. So I don't know if you want to talk a little.
Yeah, that's great. On the conjugation kit front, I'll actually follow up with that. I have a schematic actually at the end that shows we partnered with someone to do a very, very simple, very fast conjugation kit, right? There's really 3 steps where we activate the antibody, add a single-stranded oligo, and then combine the phyton onto that. Vetted a number of different conjugation kits, really found one that we like from both a yield point of view and also an ease of use. Really the goal that we had at the outset was we want to be able to ship you a conjugation kit and you could be working with these labels on your antibodies within a day. So that's the conjugation kit. Why would you want to run 40 colors? There's actually a number of different cases that this fits, right? So the first one, as you mentioned, Seddon, is for rare samples, right? We have one blood draw, you're only getting a small aliquot of cells, and you need to be able to answer a lot of questions of that small subset of cells. I would also say, you know, there are a number of different experiments, you know, that I've run where I'm looking at multiple different cell types in multiple different panels, right? Or I want to actually run multiple different panels with different activation markers or different checkpoint inhibitors. And, you know, it's very difficult to say, okay, well, that one cell that was in that sample is, well, it's the same cell that's over here. I'd really like to do all that in one tube, right? And frankly, that does save you on overall antibodies. So the reason to go high parameter can also be economic, because you can just run panel and you can do both broad and deep immune phenotyping. Cool. Send anything to add on that latter part around 40?
No, I think that's good. And then there's a question about where we are in terms of looking at other laser lines. So do we have any plans to come up with Nova Red fluorochromes? Yes,
absolutely, we do. And we'll be saying a lot more about that in June. So we're very actively working on new labels. And so that's as much as I can say right now. I think the important part of what we'll cover today will be how do we think around label performance in terms of when we think about creating a new fluorescent label. We are specifically designing to have the characteristics that I'm showing you here, right? Where we think around cross-laser excitation as a foundational item, right? We think about that tunability as a foundational item, right? And so we really want to continue that notion of stacking no matter where we go in the spectrum, no matter how many labels we create. And I'd also say, you know, the ability for this platform to create a large number of fluorescent labels very quickly is unique. And that's something that gets me really excited. I think, you know, I framed it for Seddon when she started with us. I said, you know, this is kind of like the kid in the candy store, because now you can imagine having a number of different labels. And the interesting thing about for what it's worth, is that the more of these labels that you use, there's an inverse correlation. The more you use, the less, the easier experimental design gets, because of the cleanliness of these fluorophores and honestly, the easier the data analysis and the experimental planning gets. So I think that's really, really interesting. And this is a great segue actually into this slide, because of that notion of what is the performance of these. This is run on an Attune for what it's worth. Actually, all the data you'll see today is off of either an Attune or Aurora, right? So there's no magic. We always ran with default settings. We're not using, you know, 2-watt, 1-watt lasers to light these things up, right? But I think this gives you a sense of the performance. Right? You have Nova Blue 530, which is essentially a fantasy dye, right? It only emits where we want it to. 610, really well performing. 660, Nova Yellow 570, again, another really nice dye in terms of it only emitting where desired. 610 and 660. And again, we'll take all these labels and I'll show you how they perform really when we push that envelope. So one of the things that, you know, we can think about, right, is in terms of this performance is, what is this major problem with respect to where we are in the BYG, right? And the issue is that these PE and PE tandem dyes, right, are excited off of both these lasers, as you guys well know. But if you actually look at manufacturer recommendations, right, and this is actually looking off of the Fortessa or the Attune, right, it's actually that you see these labels coming up, right, in both, you know, in both different places, really because there's that much level of cross excitation. And what this does is prevents you from using honestly the latent capabilities of the instrument, right? Because you see this coming up in both of these different detectors. And that's not the way we're doing this, right? So we can go in and stack these instruments. So while we'll spend a lot of time up at the very, very high parameter to your question around 40 colors, and while that does give us honestly a lot of insight around, well, what is the spreading impact, no matter where you are in the spectrum, I think the important thing is regardless of the number of colors that you're running, you certainly want to leverage the capabilities of your instrument, and you certainly want to have it be an easier time to plan that experiment and analyze it. And we'll show you what that means in a moment. So what does that look like? So if we just take a step back and look at just PE dazzle, for instance, right, which, you know, is a great dye in terms of its brightness, but is an issue because of, well, what's the cost of doing business when we use PE dazzle? And the answer is the cost of doing business is this incredible amount of cross excitation off of the blue. And you can see where Nova Blue and Nova Yellow come in is actually separating those and then stacking them. I think probably the simplest slide that I have in this deck is actually this next one, but I think it also gives you the kind of, if you will, the nail in the coffin part of what is the dye performance and why does that matter, right? And these are some plots, some high resolution plots of compensated data that's off of these Nova dyes, is generated by Ian of FlowJo. And I think they give you a really interesting indication, not just because, oh, hey, look, we can perform this beautiful compensation where the medians line up, but note the lack of spread. You're not getting that massive trumpet effect, right? Made a joke yesterday. I don't know if it was funny or not because most people are muted on the webinar, but we can really make quad gates great again because really there is very little spread of these labels into one another. So not only can we stack those blue and yellow green laser lines, but we're again making the data analysis easier. This is really what we're trying to drive towards, is that ability to do much better data analysis, right? And have these clean fluorophores really help drive that from an infrastructure point of view. Another part is how bright are these things, right? And which is a question we get a lot of. And our answer to that is, well, how can you separate biological populations? And the way we're looking at that is through separation index. Separation index, not stain index, because I do want to look at labels through the point of view of, well, how much spread are you getting in the negative, which separation index enables us to do by looking at the 84th percentile of the negative, right? Because you really should be thinking around what does that separation look like versus how wide your negative population is. And with these two labels that we actually got to the NIH for performance testing of the 6 that we have the same separation index as APC. So I think, you know, really nice performance on it in terms of cleanliness, but the ability to do biology. Okay, some brand new data on consistency and stability. Honestly, very interesting story for us because we went out and said, oh, we got these new colors. And the huge pushback we got was, okay, do I have to put them on my samples 15 minutes before I stain? How stable are they? Can I leave them on a bench? How far can I push this? Okay, let's answer this question. So the first one is, although obviously our light labels, like anything that we're using in the lab, like to stay cool, like to stay in a fridge, we've actually left them on a bench, again, light protected for up to 3 weeks, and there's been no change in the fluorescence. The other thing that we did is to push that even harder, right, is take this and actually fix the cells, right? So we took anti-CD3 that was conjugated to one of our Nova Yellow dyes, stained the cells, right, and then left them for 2 weeks, and then compared those to fresh. We didn't think that this was going to be a COVID relevant experiment in terms of, okay, well, now we can fix our cells and then maybe run them 2 weeks later when we get access to the cytometer, but it turns out to be, and it turns out there's actually no change in the fluorescence, right? So you can push this very, very hard. For what it's worth, these past 2 experiments, this is just the longest that we've tested, right? And the reason we think a lot of that is occurring is because we have this DNA structure, right? And it actually, instead of it laying flat the way that I showed you in the schematic earlier, it actually has this cup-like structure and provides essentially a solubility cage that really stabilizes the component dyes, both from a spatial point of view, but also in solution, right? I think that's an important part of when we think around, well, how does tandem dye performance look like versus something like a Phyton, where the labels are, the component dyes are really rigidly and stably held. Mandatory dog barking in the background because everyone's working from home, so I'm glad we checked that box. The other part of this is we have a goal, which is also we're really aiming to make lot-to-lot consistency really a distant memory, and that sounds like a crazily ambitious goal, and I agree. But what does that mean? And it has a couple of different components, right? And then we'll come to this slide in a second. The first one is, you know, we're relying on ISO 9001, manufacturing facility. The second part is that we then anneal and create those phyton structures, right? Then we actually look at, okay, well, how does that look like lot to lot? And the final, which is what we have here, and then the final one is actually that we QC on cells every single time. So every lot of fluorescent labels that leaves our facility is QC'd on PBMCs, not a cell line, not something artificial, on PBMCs, right? So that when you actually see a data sheet from us, it's exactly that lot run on cells. This focuses in on this middle component, which is, okay, when we're assembling these labels, what's the lot-to-lot consistency? And the answer is, you can see that it's essentially near zero for all of these labels, and NovaBlue 530 comes just under our spec, which is under 5% lot-to-lot variability, right? And that's just looking at this, in a spec plot. So pretty amazing. We look at this for what it's worth in flow, right, which is the business end of this. The CV of our MFIs of all labels range between 2 and 4%, right? So the lot-to-lot consistency and the ability to leverage these in longitudinal studies, we think is really interesting. So last part, which is controllable brightness, right? We really want a volume knob. How can we leverage this platform to tune up and down our brightness? Turn up brightness to get low antigen density, maybe tune it down, because sometimes that's not everything. So what I've shown you, all the data that I've shown you essentially to now has been actually based on these 2 cluster labels, and that's what we put together with the conjugation kit that someone asked about earlier, that I'll spend a little bit more time in terms of what that workflow looks like in a moment. But we built up to that by going from 1 cluster, and I'll show you because we could replicate the network, to 2 cluster, and now we're moving all the way up into 4 cluster labels. Each one of these arms of this cruciform is a cluster. So the first thing we had to show was, along this, if you think about this phyton being essentially symmetrical on the diagonal axis, that we could replicate the networks, right? And I think that's just an important science experiment to do. For what it's worth, it gives you just an interesting indication for what it's worth for the fluorescence geeks in the room and who isn't, is that when you dial this distance in, you can actually small changes in distance can actually have an interesting impact on limiting the cross excitation. For what it's worth, this gives you an indication of how we're able to limit that cross excitation, not only just through component dye selection, but also through the geometry. But that replication also enables tunable brightness. So in fact, we can stoichiometrically, in this case, tune from 1 to twice as bright. Yes, it's a spec plot. Show me the flow. It's exactly what I said when I first saw this figure, and that's what we did. So this is the NovaBlue 610 dye going from a 1 to a 2 cluster. This is a fluorescence -1, right? The fluorescence minus outcome is always how I like to think about that. Label only, where we're doping in the 1 cluster and the 2 cluster labels, and then we're looking at this in flow cytometry. And this plot, I think, reveals, although it's like, okay, it's just a boring histogram, there's actually some interesting stuff in here. One is, yes, it is in fact twice as bright. So 1, we can stoichiometrically tune the brightness. 2, I think what's important is that you are literally increasing the separation index without any change in the background. And I think this is a really important point, right? Is that we are making labels that are brighter, but they have a controlled amount and limited amount of background. I think it's really important when we think around, hey, could you use multiple of these in multiplexing, and how would I map this into low antigen density markers? But let's also push that a step further. So what we did is we took our highest separation index label, which is this Nova Yellow 610. It's kind of the star in terms of its brightness as measured by separation index, and really turned it up to 11, right? Went from a 2 cluster label to a 4 cluster label. And that's exactly what we did. It turns out to be 1.9 times as bright when we look at it on a spectrophotometer. And I'll show you what the results look like in flow now. And so this is our comparison of a 2 cluster label and a 4 cluster label. It is, as I've mentioned, it's nearly twice as bright. It's 1.84 times as bright when we look at it by flow. And here we use median fluorescence intensity actually as a proxy for how does this compare and how does this perform versus something like PE, where there's a variation of maybe 1 or sometimes 2 of these on an antibody. And what we see here is that, 1, yes, in fact, we are getting nearly twice as bright. But I think what's notable here, when we contrast this with PE, is what it's doing in the just adjacent detector. And again, this is all run on an attune, is there's very minimal fluorescence that it's even putting into the adjacent detector. And I think that's really important because we can think about going after low antigen density targets with a high brightness fluorophore and possibly even doing that in adjacent channels. So we're currently in beta on this. There's a number of different people that, we're scaling the manufacturing part of this now and deploying this. We've got people who are really interested in EV detection and triggering off fluorescence. We've got a partner of ours that's looking at this for PE and PE conjugate replacement for undetectable drug targets that they have now. There's a number of folks who are also looking at, hey, can we do simultaneous detection of multiple checkpoint inhibitors? right? Because we've got low expression of multiple, and that actually gets to your question earlier around like, why would you want to do 40 colors? Well, maybe you could do multiple low antigen detection at the same time, even possibly even do it in adjacent channels. We've also had a lot of recent interest, as I'm sure some of you can imagine, also in ACE2 detection, where they want to take a very clean or very bright fluorophore and put that onto the anti-ACE2 antibody. So I hope I've shown you those 3 requirements and really how we meet all of that said. And maybe it's a good time for a break for questions, and then we'll go into the 40 color data.
Yeah, absolutely. So one of the questions, if someone's labeling in-house, can you quantitate the moles of dye labeled for QC measures?
Yes, and that's really interesting. And yes, we've got a protocol for that, because we speak moles because we're always in the oligo world. And so the answer is yes, and we would be able to help you quantify that. For instance, on the other end, if you were saying, hey, look, on the back end of this, after you do the conjugation, do you have a way where I could measure that, the number of moles I'm getting out? The answer is yes, and we could provide the method to do that. We could also provide the number of moles that are sort of on the front end of that. I'll show you some efficiency sort of metrics later too, in terms of what the steps of those look like. For the final step of that, I think it's the middle step is like 85% efficient, and then there's 60 to 80% efficiency, at least the range that we've seen on the final Phyton to antibody binding. Because it's relying on this oligo, and you don't need to do any post purification, because you can just, there's free Phyton in solution, but that's not really binding to anything. You can wash that out.
Okay. And the second question is related to the log scale. So they're asking, how much does the difference between 1 to 2 to 4 brightness actually matter in terms of your data?
Yeah, I mean, actually, I think that's interesting in the sense that, yeah, you are on a log scale, so obviously the difference is quite a bit larger if you were to look at it at another scale. I think there's a couple different ways to answer that question. Would you be able to distinguish a 2 cluster and a 4 cluster, right, if you're trying to gate on them? I think the answer is no. Could you distinguish a 1 and a 4 cluster? Absolutely. How much is that difference? I think what we always like to shoot for, and we've seen this in some performance data that we've looked at, is we usually look at a 2 cluster labels, at least of these 6 labels, as comparable to some of the comparator dyes that we've looked at. So for instance, if you compare NovaYellow 610 in a 2 cluster basis to something like Alexa 694, right, it's actually brighter than that, or the ECD dye from Beckman, it's brighter than that, just in a 2 cluster format. So I think, yeah, again, while it's on a log scale, I think what you have to kind of keep in mind here is that you've got a very bright dye at baseline, and now you can actually move that brightness much, much higher but you're doing that on a much cleaner label.
And another question, with the Nova Blue 660 and the Nova Yellow 660, they're seeing cross-excitation with the red lasers. So, and you may want to address.
Yeah, so this is something where, so the APDs are very sensitive in the red, and the gain in the red is really high. And so why is that? And that's on the Aurora data that you're referring to, because you saw those red plots. That's so that the Cytec can distinguish. I believe it's Alexa 4. What is it? Is it Alexa 4 700 and APC 700? Anyway, it's setting. You'll have to correct me if I'm wrong. There's 2 red dyes there that are very similar. And so when that gain's amplified, you can't actually separate those dyes very faithfully. What I'll show you later, though, is that there actually is no contribution of spread into the red off of these labels. So while you're seeing that cross-excitation, that's data that Cytec produced and then posted up, you're not seeing any of that when you actually get into the 40 color panel that we'll show in a moment. And I'm sorry that I forget the labels off the top of my head that are distinguished off.
Floor 647, several people were helping to answer that as well.
Thank you.
Yeah. So,
awesome.
And then there's a question in whether we have any plans to go beyond a 4-fold increase in brightness, and would this make the Phyton a lot larger to accomplish it?
Yes and no. We'll have to pick that apart. So there's an inherent quenching limit on the Phyton itself. And we have some of our labels are actually single components. In other words, there's just single dye on there. And we can actually go much higher brightness than you're seeing here. And we know what that limit is. So we wouldn't need to increase the size of the Phyton at all to do that. Have we thought about strategies for increasing, either the size of the phyton or going for another strategy, for instance, to get a very, very high signal? Yes, absolutely. Would that come with the cost of creating a larger structure? Undoubtedly. To that point, let's get ahead on that question, which is what is the size of it? When it's laid out flat, it's 20 nanometers across. It's 150 kilodaltons. Remember the point that I made earlier, though, it has this cup-like structure. So it's actually probably more like 18 to 19 nanometers across. So it is much, much smaller than PE, right? And so let's get ahead on the next question. How about that, Seddon? Intracellular. We're testing it now. And so we're very open to saying, hey, which targets are you interested in? What do you want to look at in combination with other markers? So, and Seddon's very actively planning those experiments right now. So hopefully maybe I jumped the line on answering 2 questions there.
Yeah, no, good. And then the last question is there a bleaching effect with high laser power, so greater than 100 milliwatts?
Good question. We have run it. We've run these labels on a very high power Symphony that had lasers that were tuned up to 1 watt. Again, saw great performance of the labels off that. Have we done any experiments, for instance, where we've looked, we've run the labels, hit them with that laser power, and then looked afterwards? No, we have not done that photostability experiment. Great question. I also think, there's been a lot of conversations that we've had too around, people seeing, if you will, differences in the signatures, the difference of the spectrum after treatment like that. And so that's another question that we're really intrigued by and really want to address, honestly, within the next couple of months. Is that right, Seddon?
Yeah. Yep.
Yeah, I love that question. And I think rigorously pushing photostability is, as you can tell here, is kind of the next thing we really want to go after. Again, because of if we've got solution stability, if we've got temperature stability, if we've got this kind of consistency, is that structure also really stabilizing, the photostability? I'd say anecdotally, right, we're not manufacturing this in red light. We're manufacturing in a standard lab environment. And, there's no, again, the other thing too is there's no special buffers that we're using here. Everything you saw was stored in PBS, right? So as long as they're in solution, we're good. Cool.
Okay. Got 2 more questions. So one, I think you started to address with the intracellular, but the question is, have we tested them in phosphoflow, the stability and fixed and perm buffers? So as Mike already mentioned, we're wanting to go there actively. So if you have targets that you think would be great test cases,
send us your targets, right? Yeah. I think that's the biggest thing. We want to know what you're interested in. We haven't tested methanol fixation either. I think similar to you, we're chomping at the bit, particularly because of what we're seeing in terms of the ability to multiplex, right? And think about, where in the cell can we go? What can we light up in there? And then, is there a notion of also tuning up the brightness for some of those targets? Definitely of interest. So, I would love to hear from you guys in the chat about, hey, these are the targets we're really interested in. This is how we're doing it. And we're also doing a lot of buffer testing. You asked the question around fixed perm, and so we were doing a lot of honestly simple experiments now around buffer testing, around, does it work, what about fixed perm? What about brilliant buffer? What about, so I think I'm very optimistic on that because of this DNA structure and this notion of the solubility cage, some of the stuff we've seen with fixative. Again, we'll prove it out and we'll get the data and we'll roll that into a future webinar for sure.
Okay. So the next one is for in-house labeling. Can you control the number of clusters that are conjugated to the antibody? And if there are pre-conjugated antibodies, how many clusters would be on those? Right.
So good question. So yeah, there's two different things going on here. So the cluster, when you've got a label in a tube, right? It's NovaFluors 610, it is all 2 cluster, right? So it is all this. It's all 2 cluster label. There's not even any variation in there. It's 100% 2 cluster label. The conjugation kit is putting actually a actually very narrow banded number of those phytons onto your antibody, right? And we've actually found that we're able to get a very consistent labeling of 1 phyton on 1 antibody, which leads us down another interesting conversation path, which maybe we'll visit at the end, right? But what we're aiming to do then is to kind of extend your question. Hopefully I'm anticipating kind of where that was going, which is we can also send a 4 cluster label and then you can do the same labeling, right? We've also, I think, thought about and started working with our partner on, could you also use degree of labeling as a way of changing the brightness, right? I think that would limit maybe that limits our ability to go intracellular. We don't know. but it certainly is another modality that we've thought around as a volume nub. What if you went 4 cluster and you had a degree of labeling, for instance, that was 3 to 1, not 1 to 1?
And then the last question is, what about paraformaldehyde fixation? So yes, we've tried paraformaldehyde as well as formalin fixation.
That's right. Yeah, we tried both. Yeah, and it's incredibly stable. I mean, I think it's really interesting. I mean, I wouldn't have left it in fixative. I probably would have fixed it and then left it in PBS. That really pushes the system pretty hard, just in terms of how dyes can perform. I think, we're very much interested in seeing, one, how long can we go in terms of bench stability, and also in terms of stability in fixative or post-fixative treatment. Is there any degradation in the fluorescence? Everything I've shown you, though, is just, it shows you that it's an incredibly stable platform.
All right, and the next one is setting you up, I think, well going forward. They want to know how the NovaFluors perform with the 6 NovaFluors, dropping them into the 34 color Cytec panel.
Someone I always lean when because that's a perfect segue. So I'm going to take us into the next part of this. That's a great tee up. So whoever did that, we don't actually have any swag. I would promise you swag, but we don't have any. So we'll just have to hit you with, all of this great data. That's what we can offer. And so, yeah, this is our effort to go above and beyond 40. Great number of resources here as well, right? And so I'll start off by all this and try to give you the Lamar Burton plug here, which is that, I'm going to show you a lot of data. And I'm going to show you how we designed this panel, how we thought about this panel. Don't take my word for it. I will quite literally, after this webinar, be posting up our data, both hopefully on Flow Repository, if the server can handle it, and also on our GitHub site. And I'll show you the links in a moment. You can take our data and you can look at it. You can analyze it. We'll also be sharing the FlowJo workspace where we analyze this data. So that's part of this as well, right? Is sharing some of this data. So not that you can tune me out because you're like, oh, Mike doesn't know anything about data analysis, but point is you can also grab this data for yourself and perform some of the similar kind of analysis that we've done, or hopefully extend it, because as I'll show you, there's some interesting biology, I think, to be had in this data set as well. So what do we do? So we base this on some beautiful work from Cytec, where they had this 35 color panel. We got 34 of the commercially available ones and dropped CD19 out of that because we just weren't able to get it. And so we started with this 34 color backbone panel, and the details of that panel and running it on the 5-laser Aurora are certainly available on their website. And that's really where we started. That was the seed of this. And what we did to test this and really push this notion of, hey, what about stacking? Can you just simply plug and play these labels? That's exactly what we did. So we took the 6 labels that we have now and plug and played them into the blue and the yellow green. We'll talk all around the performance of that in a moment. But one point I want to make as well is, we didn't put all these on CD4 or high antigen density markers, right? We also put this on chemokine receptors. We put it on CD40, or we put it on markers where, to the point earlier around sort of brightness and how can we work with this, put on markers that honestly drive some interesting biology. And I'll show you some results of that in a moment as well. And that's what got us to this 40 color panel. This is the busiest plot that I've got on maybe the whole deck. But the point is you can see, because we're hitting those detectors cleanly, that we can literally just drop these in. It has some interesting effects too, for what it's worth, particularly on other yellow-green excited labels. What we did too is, again, because we can actually look at the similarity matrix of dyes, we actually took the 34 color backbone and just looked at the similarity matrix of that. Look at the similarity matrix of our 40 color panel in terms of, okay, you're dropping in these 6 labels. What are they similar to? Can we anticipate any issues? And of course, you get the similarity matrix out, but anytime I see a plot like this, of course, it's almost like a reflex. I have to cluster it, and so that's exactly what I did here. And so what we're actually doing here is we're clustering labels based on their similarity, right? Based on this similarity of dye versus dye. And so this leads us down a couple of interesting paths that I think will inform the discussion as we move forward. The violet dyes display bunching. In other words, there are violet dyes that are very similar to one another, and this manifests itself in the biology, right? When you're trying to look at something like BV480, which was on the IgD, versus C3, which is off the violet 510, right? So there's some impacts of this, because even though those are different cell types, you have very, very similar dyes, and that can lead to some interesting data analysis there. The other one, which is honestly anticipated, is that unmixing BB515 and FITC is challenging, right? I mean, the similarity of these labels is very, very high. For what it's worth, again, very transparently, adding the Nova Blue 530 to this makes it worse. And that's kind of what you'd anticipate, right? It's in that same ballpark. It's also that none of these labels really have much in the way of a signature, right? Well, our recommendation here, again, to leverage the notion of hitting those cleanly is really is to use Nova Blue 530 with either Brilliant Blue 515 or FITC alone, right? And for what it's worth, that actually introduces less complexity into your panel design, again, as measured by this complexity metric, you know, that you can generate off Cytec's website. PE Dazzle, I mean, no surprise there, right? PE Dazzle is this problem child, right? It's very similar to it's got a very high similarity index versus, you know, compared to a lot of other labels. We'll actually touch on this in a moment, though, when we talk around like what does this look like in terms of spectral spread, because that's actually very impactful. The other one is a final, which is sort of a, if you will, a firm recommendation, which is why would you just not replace PE Sci5, right? Which is this not very well performing dye, particularly on other cytometers where you might not be able to pull apart PE Sci5 and NovaYellow 660. Really, NovaYellow 660 performs as a great replacement for that, right? Much, much better, in fact. So another way that we try to visualize this is what is the primary detector for these dyes that we've got, particularly in the BYG space, and what does putting one of our labels in at a time do to that, right? And then we'll also look at spectral spreading and how that looks like as we add one label at a time, because that obviously impacts data analysis. But I thought this was interesting from the point of view of like, how are we leveraging these detectors? And sure, we're looking at signatures, but I think it's also interesting to look at this from the point of view of what is that primary detector? And so per CP, right, that you've got, you know, presumably it's going to have its peak fluorescence actually in blue, and it's going to be detected in this BA channel. But in fact, what you find, right, is that its peak fluorescence and where it's detected is in the YG5. And so we'll use this visualization to actually step through taking this 34 color backbone panel and dropping in one NovaFluor at a time. And so what we see is Nova Blue is detected, honestly, exactly where you'd expect it. You've got, you know, Brilliant Blue 515 hitting and FITC hitting the first two blue detectors. You've got Nova Blue 530 that's hitting the blue. And so that's essentially as desired. Again, Nova Blue 610 hitting the B6 detector as you'd expect. Similar story for Nova Blue 660, right? We see a bit of a shift in here where it's hitting the B8 detector, but we're hitting all three of the blue detectors very cleanly. And again, hold the thought around spectral spreading in a second. This is just the notion of essentially just the primary detector for these labels. Then interesting things occur in the yellow-green, right? So when we put CD19 onto NovaYellow 570 and we can hit that YG line, we hit that YG detector so cleanly, we actually essentially push PE's primary detection into the blue, right? And so we can actually go in there and stack, leverage all these together, no doubt, right? That's how we're able to get to that level of panel. But I think it's interesting because when you do hit that detector very cleanly, if you will, the primary detector for another label moves. Same thing for NovaYellow 610, right? We hit this YG line, we actually push PE Dazzle out to be now detected in the blue, right? And then another similar story here where when you use NovaYellow 660, because again, it's hitting that YG5 detector so cleanly, you push PE Sci5 out, which is interesting. So but hold this thought because we need to talk around spectral spreading because that considers really this on a whole spectrum basis. This is just all of this measured together. Sort of the upshot is we can hit the blue detectors very cleanly, and then we label it when we layer in these yellow detectors, we're essentially pushing these badly performing YG excited dyes to be detected out into the blue, at least for their primary detection. Now let's focus on looking at this on a whole spectrum basis, a much more, I think, honestly interesting and more fair way of looking at this. And so what we did is we ran the SSM, the spillover spreading matrix of the 34 color backbone. You know, you see things in here, honestly, that you'd anticipate where you've got some interesting performance where E-Fluor 450 is spreading quite a bit. You've got a lot of spread BUVs, the per-CPs, again, as you'd expect off of the blue, are adding quite a bit of spread. And what we did is ask a question of, now that we've got the 34 color, if you will, backbone panel, when we add one label in at a time, what is the change in spread? And so the way we did this is actually take the spillover spreading matrix of the 34 color backbone and literally just did the delta of the SSM versus each one of these drop-ins. And you can see, well, what is the impact of adding each one of these labels? And so we'll walk through each one of these. So on the y-axis here is just that delta of the SSM. What is the change in spillover, right? So what you'll see here is spillover spread is, so when we drop in just the Nova Blue 530, it performs honestly exactly how you'd expect. Note the y-axis here because it is quite large. Is the spillover spread is specifically increased for BB 515 and FITC, but then no other dyes are affected, right? So this, again, a spillover in those, for those labels is high, but there's no impact anywhere else. Little to no change for dropping in the Nova Blue 610 or Nova Blue 660. So essentially marginal impact. And this is that notion of if you have low impact on spread, you can plug and play these. And then consequently, of course, data analysis should be a lot easier. NovaFluors 570, honestly, what we'd expect. We see, and note the axes here are not the 2000 we were seeing in Brilliant Blue 515 and FTSE, right? Small changes in PE and CF 568. We see one small change in the spillover PE dazzle when we drop in NovaFluors 610, and we see a small change in the spillover PE Sci 5 for 660. Honestly, I think what's important about these plots is there certainly are individual dyes where we're having small impacts on the on an increase in spillover, but I think it's what you're not seeing that's what's important. And that is we're adding essentially no appreciable spread anywhere else, right? And so we're hitting these very, very cleanly. And again, you can still distinguish these dyes. It's just the spillover is increasing. So what's the TLDR here, right? You're like, we're not enough work, Tessa. Like this is crazy. Why would you push this? The point of this is that we've added 6 colors with marginal impact on spread, except for one that we anticipate because they're very highly similar dye, and near zero spread added to other channels. And the impact of that really is easier experimental design, higher resolution of these biological populations because it means less spread, and easier data analysis, right? You're trying to cluster on these, you want clean data, right? So we also work to make this honestly understandable, right? And so this is our subway plot visualization, something I wish I had honestly thought about years ago when I was presenting some of these larger data sets. And this is just how we can navigate the immune cell populations. And I think it just gives us also a very transparent view of like, well, how did we gate this and where do we go on that? And then also how can we look into this data and peer into it much deeper? This I know is an incredibly busy plot. Some of these plots are incredibly small. Again, we're sharing all this, and all these individual plots are going to be available up on both the GitHub site and Flow repository. But I think what's interesting, just to kind of call out some things, is again, I mentioned earlier that we're using these NovaFluors, not just on CD19, but also on some of these chemokine receptors. And of course, what that enables us to do is peer in and subset very deeply into these populations for chemokine receptor expressions, for instance, on the different B cells here, right? In this case, we're comparing 4 different B cell populations for CD40 for CXCR4, and we can really peer in there and use these now to drive deeper insights and deeper discoveries. And again, to that point, actually, we obviously ran some advanced analysis on this, right? We're looking here at the coloring we just used in the subway plot, and used UpSNE to just perform this T-SNE projection. And you can see here, for instance, that the color I've added, which is gray, shows that there's a lot more. I mean, there is, at least based on this visualization, and it's a visualization only, is that there is some population diversity in here, right? And we try to pull that out in FlowSum, which I'll show you in a moment. But there's a lot to be discovered in this data set, right? And we think a lot of that, it's driven out of the point of you have very high resolution that you get of some of these phenotyping and some of these markers that you've got laying below the phenotyping surface. To that point, I mean, you can see, for instance, we're lighting up the B cell population with CD19, but you can really see the variation in some of these chemokine receptor populations, right? When we perform this color mapping. Again, we're sharing all this data, so this is a very top level view on like, hey, what's possible, and we really want to also sort of leaning, if you will, on the field to say, look, what are you seeing in the data? Are you able to see things that we didn't or just haven't gotten to the depth on this? Similarly, we see a great number of diversity of clusters in FlowSum, right? 18 meta clusters and obviously many subclusters to that. And again, this is, Ian generated this plot for us, and I think gives us a sense of, particularly leveraging some of these tools or leveraging Cluster Explorer, how you can actually dig in and discover some new biology in this data set. With that, what are we doing with it? So the white paper is available actually up on our website, or you can email info@phitonex.com or me, and I'll give you my email address in a moment, where you can get that white paper. It provides the methods and materials for how do we do this, how do we run it. It also shows a lot of the data that we showed and a little bit more in terms of panel design. The data set also, this page has been stood up, but I actually haven't had a chance to upload the data yet because yesterday was a very interesting day. In terms of the level of interest in the 40 color panel and what are those capabilities. And so I'll be uploading that data hopefully today, but certainly by the end of the week, where we'll have both the FlowJo workspace as well as all of the FCS files that drive this. I'll note too, it's not just the 40 color data, it is the 34 color, the 34 plus ones, and also the 40 color data. So all the different things that I was showing you today. And then also hopefully up on Flow repository. So then, really the kind of the final question, if you will, and then we'll open it up and have a discussion. How do we do this? How do we get access to this? So we've packaged up the 6 labels into a CD4 test kit, something that, looking at fluorescent labels and characterizing an SSM on an instrument, something I've been a huge supporter of for years now. Exactly what we wanted to deploy there, right? So you can look at the CD4 testing kit, look at that on human SK3 and characterize these labels so you can drive it into your own panel design. The second thing is, and something we've had quite a few questions around, it was this NovaFluor conjugation kit. It's a 6x100 microgram conjugation kit. I'll actually show you the steps on the next slide. That also, so that gives you that ability, again, to conjugate up to 100 micrograms of each of 6 different antibodies to the labels. And then we've also supported a number of different custom conjugation projects. And so this is where someone says, look, I want, for instance, in this case, NovaYellow570 on CD45. Can you please conjugate that for me? Sure. So milligram and up, happy to do that and take those on and have that discussion with you. Diving just a little bit deeper in the conjugation kit for a moment, and then we'll wrap up and have a discussion, is what does that look like, right? So I mentioned 3 steps. So the first one is you've got to activate the antibody and then add this ssDNA linker, right? And honestly, very simple, right? You adding antibody to a reagent, you incubate it, right? Leave it overnight. The next day you're removing the ssDNA, you're spinning it, and then you add on this Phyton. And it's this complementary sequence that really gives us a great efficiency on this side, doesn't require us to post-purify. So again, someone had asked earlier around molar yield, 85% yield here, and we've seen anywhere from 60 to 80% here. We're working with a partner. They have tested thousands of antibodies and have never seen any loss of specificity of the antibodies, which is quite impressive. And also this is kind of part of our push to really say, hey, look, conjugation can also be honestly quite simple, and you could get to the point where you're running these in a flow experiment within a day. So hopefully, I've shown you really 5 different things here. We've got labels that are bright and clean. They're consistent and statable. They're tunable in terms of both their excitation, emission, and brightness. They're also plug and play, and I think that's an emergent thing, which is that you can upgrade your current panel really regardless of the flow cytometer you're running on, right? We obviously pushed it to a limit to really see how does it interact with a number of different dyes, but it applies across a number of flow cytometers. And really you can discover more, right? You can think about fluorescence for biology, not dealing with it the other way around. And hopefully maybe, and I think it's ambitious, but a new way to think about panel design. So if you're interested in getting the white paper, please email me, mike.sednisky@phitonex.com or info@phitonex.com. And would love to have a great discussion around all the different questions you guys have and keep that going.
All right. We've got one question, which I don't think I've even thought about. In addition to antibody, can we also label cells?
Can you label cells? Yeah, I don't see why not. It's just a protein labeling kit. Good question, and it would be, I honestly don't see why not. I think the question would be, what would the stoichiometry look like from mapping, if you will, if you got a kit that's sort of targeted for 100 micrograms of antibody at 1 mg per mil, can you map that into a concentration of cells? And do you have enough, essentially you should have enough free lysines on the surface to conjugate. So yeah, it'd be interesting to map out how many and what the stoichiometry of that reaction would look like. So have we done it? No, like straight up, no. We haven't done it. Do I have data? No. Could we figure that out with you? Yeah, I think that'd be really interesting.
Yeah. Okay. So the next question, so does the DNA of the Phyton act as a mop to clean up the emission?
No, it does not. It's an interesting question. You guys know the absorbance of DNA, right? Everyone's done sort of these absorbance experiments. Obviously we're not playing in that. It does not act as a mop. Which, sorry, I was distracted thinking around literally someone with a mop that was made of DNA and sliding it. But no, it does not act as a mop. It is acting purely in a structural capacity. And the nice part about having DNA, there's a couple of different nice parts of it. Is one, when you do the sequence design, you're able to control that spatial distribution very consistently, right? Like when I said like there's a when there's two cluster label, you're only going to that two cluster label, you're only going to two cluster label, right? And the distance is always the same for every single label in between the component dies because of the sequence design. The other part of it, of course, is this notion of annealing, right? Is that those structures are self-assembled, right? And then the third part, which I think has been a really interesting part for us, has been this notion that you can make it a very flexible platform in the sense that you can hang a single-stranded oligo off of the end of this. And that gives you a lot of flexibility, right? Beyond just labeling a protein or an antibody, or as you asked, a cell or something else, right? Because you just have a piece of DNA hanging off. And we, I think many of us could think about some interesting ideas about what that could be used for.
All right. And then there's a question whether it can be used in whole blood applications.
Can it be used in whole blood? Yeah, so we haven't tested whole blood. We have had a partner that tested whole blood. In that case, so we have a blocking solution that we ship with these kits. The blocking solution will be really important just because of the stickiness of monocyte macrophages and other cells binding up to the NovaFluors. It's the same thing that they do to some of the Cy-based dyes. So if you've ever worked with Cy5, Cy7, and you've seen some of that background, I would just recommend that you probably use maybe a higher dose of block if you're using that whole blood. We've not gone back and tested that whole blood with the essentially new formulation of this block that we've done. That's an experiment, that we've got to do. But yeah, did they fluoresce and perform well? Yes. Was there high background when we tested that pre-block? Definitely. So I think we've got to get back in there and if you will, get under the hood and treat probably with a higher dose of block. For what it's worth, probably worth a mention is that the blocking solution, when Saturn was performing these experiments, it's a 5-minute incubation step at pretty low volume right before staining. Do I have that right?
Just add 5 microliters to the block.
Yeah, so we'd probably maybe recommend a higher dose, right, for whole blood pre-stain.
Yeah. All right. So another question in terms of the antibodies in the conjugation kit, do they need to be of a certain purity, similar to the MaxPar antibodies for Cytoph?
Yeah, I think the big thing that we've seen here, Seddon, right, is no other protein, right? So we recommend that the concentration is at, I think it's a mg per mil and no additional protein in there. Other than that, I think that's really our only recommendation. Do I have that right? Yeah.
Yeah, and even you can, do it with a lower concentration, either concentrated or you might have not as good of a conjugation, but that's the no other protein is an issue just because you'd be labeling that other protein. So, all right. Would the Nova block be helpful for labeling other dyes than the Nova dyes?
Yes. I'm excited about that, not because my baseline excitement levels sort of 10 or 11, but also because we just tested it versus another monocyte blocking solution, and it performs essentially as well or better, depending on the conditions, for other Cy-based dyes, Cy5, 5.5, Cy7, etc. So we went in and did the work of characterizing a receptor on monocytes and macrophages that was binding to what those labels were binding to, and created our own proprietary solution for doing that. So yes, absolutely. And I think even regardless of, hey, I'm not ready to use these dyes or whatever it is, the block, we're happy to send you block as well.
All right. No other questions right now. So
cool. We'll hang out for a couple minutes. I won't subject anyone to any more royalty-free music. But yeah, if you've got any additional questions, please hang around. Be happy to answer those.
So we've got another question. Have you observed any cross-reactivity with the Nova dyes similar to that of the Brilliant polymers?
No. Good question too. Yeah. No. Yeah, and I know what you're referring to here, and the answer is no. There's been no cross-reactivity. We've done a lot of, we're doing an increasing number of sort of master mix studies in terms of the stability of that. But we did one when we created, for instance, a TBNK kit and left essentially let them all incubate together. It was for up to several days or a week. I don't remember the time frame, but we weren't actually even looking for the reactivity. We were actually looking for, okay, based on this oligo interactions, we're getting any exchange in solution. So two things: one, we didn't get exchange in solution, but two, there's no reactivity issues. I'm looking at the Q&A now. I'm realizing that someone asked, why would you want to do 40 colors? But then I think the second question was sadism ? I think, just to comment on that, I think the reason to go that high too, even if you're not going to 40 colors, is to look at the impact on multiple different dye families. Because I think regardless of the cytometry you're running, I think it's really important to figure out what is the contribution and look at that, because someone could be running 3 in the violet and maybe they're only running 18 color panel, or I think it gives you, when you're pushing the envelope, it gives you the ability to actually assess all of that. So Seddon, is that the way you think about it?
Yeah. I mean, I think it's that, and yeah, every once in a while you just have that sample that you're never gonna get again, and that's the other.
Yeah. And that gets, I think that leads us to an interesting place, right? In terms of like, how many colors could you do? Right. And I think we've got a lot of questions on that as well. And I think that's going to be exciting to see. Great, cool. All right, well, I'll wrap this up. The recording will be posted up on YouTube very shortly. And yeah, thank you guys. And honestly, really appreciate all the messages. It's, as you can imagine, it's just been an awesome amount of work. We've had lots of fun, but yeah, really appreciate the messages. So it's great, and look forward to talking with you guys individually about this.